Hello and welcome to the next aspect of carbon cycle, that is the microbiology of starch degradation. Starch is one of the most important polymer when it comes to reserve food material in plants. As we saw in cellulose, the cellulose becomes a key component of the cell wall of the cells. Here starch is taken inside, that is photosynthesis which results, and the main end product which is starch. As you can see here in the figure, the light energy which is there, it is converted into sugars directly by photosynthesis, the process which we call as respiration or the photorespiration. So once the sugars are formed, that is glucose molecule is formed, it is attached by bonds and monomers are converted into polymers. Sugars are stored in various parts of the plants, like you can see it is stored in the leaves, in the fruit, in the roots, shoot, stem, everywhere. If you take the case of potato, then starch is stored in the tubers. So this is two-step reaction, that is assimilation and then the microbial dissimilation, which takes place when the plant litter is fallen on the ground or the dead plant material is there, or whatever the starch which gets added in the soil. Various mechanisms are there by which assimilation and dissimilation takes place with respect to carbon. So once again I would like to throw some light on the carbon cycle, as we have been seeing this cellulose and starch degradation with respect to the carbon cycle. As I have been emphasizing that the life on earth is based upon carbon. Carbon cycle is a set of reaction, a set of processes which governs the movement of carbon elements. Whatever carbonaceous compounds are there, they move through the living organisms into the environment, and how this carbon is stored in various ways. As you can see, the major stores of carbon all over the earth, rock carbonates, they are the maximum. If you see this size in gigatons carbon, size per gigatons of carbon, rock carbonates are the maximum, and most of them is trapped in the atmosphere in the form of carbon dioxide. But that carbon dioxide, if you see here, atmospheric carbon dioxide is a result of respiration or fossil fuel burning, the vehicles which we run, volcanoes, etc. I have explained all these things in the introductory aspects of carbon cycle. Here the main focus of study is the cycle which we are trying to describe in terms of stores of carbon and the processes which govern the exchange of this carbon with the environment and with the storage. Let us call it as a sink. Means what is a sink? Just like the bathroom sink, input is equal to output. As long as this is going on well, the carbon sink is maintained. The carbon sink needs to be maintained because all the life processes on earth which is there and the climate change, that is climate changes on the surface of the earth, the atmospheric change, is all governed by this carbon cycle. When we talk about global warming, we are talking about the carbon cycle. Try to understand. So you see here the focus of this polysaccharide degradation, whether that is cellulose or starch, is from here, the living biomass. These living biomass, they trap the carbon via CO2 fixation, that is photosynthesis, and a significant amount of storage it becomes, that is the plant biomass, it becomes a significant storage of carbon with these processes. So this is how starch plays a fundamental role, or you can say a substantial role in the carbon cycle. Now if you see the components of global carbon cycle, where cellulose fits in here, where the starch fits in here, you can just decide. See here, this is the land, atmosphere, and the land and the carbon dioxide interaction, whereas 70% of the earth is sea. So atmospheric carbon and the sea interface, surface of the ocean, there also there is interactions going on. So what are these interactions? You can see on the land as well as on the sea, we will see it separately. On the land, photosynthesis, plant photosynthesis is a major reaction going on. plants, they convert the atmospheric carbon dioxide into organic compounds to build the plant biomass. Out of this plant biomass, cellulose and starch, they are the key carbonaceous compounds. Therefore, we are trying to link this carbon into the carbon cycle, that is the polysaccharides to the carbon cycle. The second important carbon which is there, carbon dioxide in the atmosphere. Carbon flow, how does the carbon flow in the plants? Regulatory networks and biochemical pathways, they control how much organic matter is to be fixed and how much organic matter is to be released as carbon dioxide, okay, from the process of respiration. Now this is the carbon flow in plants via carbon dioxide fixation and then release, substantially degradation and release. So above the ground and below the ground, we have seen these things happening. Below the ground, if you see the role of microbial interactions, the rhizosphere, chemical exchanges, that cationic exchanges between the plant roots, the soil microbes, the fungi, which influence the nutrient and water uptake, plant productivity and the soil carbon content. These all act as a sink, means they store and they are somewhere involved in the mobilization of this carbon currency. Then microbial respiration and decomposition, which takes place with respect to the plant litter. That is the fourth important aspect of the global carbon cycle, that bacteria, fungi, they decompose plant litter. In plant litter, that is the remnants of roots, leaves, stems, etc., there is cellulose as well as starch. Okay, most of the time what happens, we humans, we have a significant, what you can say, digestive system to degrade starch. So we don't allow the starch to get waste as the cellulose does in nature. So we procure this starch and we take it ourselves for energy, for food. Industrialization is there with respect to starch. Commercialization is there. Starch based products we produce. With cellulose, that doesn't happen in the animal world. So most of the soil litter, it is made up of cellulose. But starch is also a key product which is formed by photosynthesis. So we have its link in the carbon cycle. Nevertheless, microbes, we humans, what we do when we eat starch, we degrade it with our enzymes, and when we defecate it, we release the gases and we release the organic matter into the environment. So bacteria, fungi, even humans, they decompose starch, I can say, and into organic compounds and release it into the environment. This is on the land what is happening. On the sea, if you see, both they have a common ground of the soil inorganic matter, or SOM. So enzymes released from the microbial transformations of the plant litter, it will involve cellulose as well as starch. They get converted into different diverse structures in the soil. Means in the soil, when the cellulose or starch, or for that content any organic matter, when it is degraded, it becomes more simpler form, glucose, then some methanol, gases. Ultimately it becomes the organic content of the soil and the soil becomes fertile. It is called as soil organic matter, which contains heavy amount of organic matter and enzymes responsible for degradation. On the sea surface, the scenario is somewhat different. Phytoplanktons and the photosynthesis takes place. Algae and photosynthetic bacteria, they form the base of marine food chain. In the marine ecosystem, the basis of food chain is the phytoplanktons and the photosynthetic bacteria, the blue-green algae. Okay, and they convert the dissolved carbon dioxide in the sea water into energy-rich organic compounds. That is, they make up the living cells. So all the life forms in the fishes, crabs, then shells, or you can say tortoise, any type of life which is there in the sea is from the conversion of dissolved carbon dioxide into life. So this consumption and respiration of these living organisms then starts. That is, they consume oxygen, release carbon dioxide again, which gets dissolved into the sea. Then one time comes when they are dead. When they die, decomposition takes place by the sea bacteria. Here one thing what happens is, whatever happens on the land, it is added into the sea, in the soil. On the land, whatever is dead, insects, plants, humans, they are added in the soil. So it doesn't go deep. It has certain layers. The layers of soil organic matter is specified, defined. But in the depth of the sea, microbes, they decompose the dead and decaying organisms to form carbon dioxide and dissolved nutrients. So what happens, a major fraction, a small fraction you can say, but it is a significant fraction which goes at the bottom of the sea, it sinks, and there the carbon is trapped. So this is the global carbon cycle in general. It was just a recap, and you can see here the carbon dioxide, when it is getting fixed into plants, when the plants they form starchy materials like fruits and vegetables, and even in roots, leaves, some of the starch is stored, it becomes fixed, that is a carbon sink is created. What is a carbon sink? The carbon sink, if you see, just it is just like the sink or wash basin. Just you think about, this is a faucet means a tap. This is a tap which is adding water, and there is a hole which is draining water. If the size of the faucet is equal to the size of the drain, means whatever amount of water is being added, that much amount of water is being released, input is equal to output, then what I mean to say is that the input is sources of carbon, that is fossil fuel combustion, deforestation, the decomposition, all this which becomes the sources of carbon. Okay, and what is the sink? The sink is uptake, that is the release of the carbon dioxide, which will be then fixed by the plants. Okay, it is in a cyclic fashion. So the if input is equal to output, then the carbon sequestration is proper. What is carbon sequestration? I will come to that. This has to do something with the carbon cycle. So here we are talking about the photosynthesis taking place, getting trapped in the form of fruits, vegetables, leaves, roots, shoots, etc., and then degrading, getting degraded, and soil organic matter, and then the whole thing released back into the environment in the form of carbon dioxide. So photosynthesis, storage, release, photosynthesis, storage, and release, this which is there, this, let us, if you just imagine it as a sink, then reservoirs which retain the carbon and keep it away from entering the earth's atmosphere are known as carbon sinks. Means what is this blue sink trap here? That carbon is trapped. When this cycle is going on, you have, let us say, 1000 kilos of potatoes. 1000 kilo of carbon in the form of 1000 kilo potatoes is trapped, carbon is trapped there. How it will be released in the environment? Either it has to be added in the soil, let the soil microorganisms degrade it, or industrialize it, make some wafers, eat it, defecate, and that will be again released in the environment. So once a starch or a carbonaceous compound is formed, either it will be stored in the form of the sink or it will be released in the environment. Same thing is there with cellulose, same thing is there with starch. What is the problem here? The problem is, in today's times, the input is much larger than the drain. Means we are releasing carbon dioxide larger as compared to the fixation. Storage is less and release is more. Okay, for example, let us consider deforestation. Deforestation is a source of carbon emission into the atmosphere. When you cut the, or when a forest is on fire, when you cut the trees and burn it or just use it for something, then that is what you can say, carbon emission. We take it in the form of carbon emission. But if a forest is growing, if a plant is growing, then that is carbon sequestration. That is the forests themselves, they serve as carbon sinks. Sequestration is to accumulate the carbon, not allow it free in the environment. There has to be a balance. Okay, we have to release also, but how much has to be released? It should not be more than which is accumulation. Right now, if you see the size of this faucet, the arrow is the size of the faucet, that is much more. Faucet is a tap, so just symbolic, that is, so it is larger than the drain. So there is an imbalance, there is global warming. So carbon sequestration is an example. This starch is very good in sequestering carbon. It is trapped in the form of plant materials. So this is, you can see numbers of how much carbon is given in the atmosphere, how much carbon is trapped, the net land sink. This figure shows the carbon sink, carbon sequestration and carbon sink. Sequestration is trapping, that is forming a sink, and then when it will be degraded, it is released in the environment. The concept of carbon sink has a very important role in the climate change. Sinks generally they act to retain the carbon, they sequestrate the carbon, okay, and only under certain circumstances they will leave it back. So stores, when you say stores, they can act as sinks and sources both. So carbon sinks are based upon the ability of the plant, in particular trees and soil, to temporarily store carbon. Soil stores carbon in the form of soil organic matter, and trees they store carbon in the form of cellulose and starch. Okay, so the quantity of carbon sequestered means stored, is in the terrestrial biomass and the soils. It is quantity of carbon in terrestrial is more, okay, then compared to soils, and terrestrial is more as compared to atmosphere, it is larger than that. So the carbon sequestration, it acts as a sink or a storehouse, and the concentration of carbon dioxide in the atmosphere, what is given is by degradation of this sink. Temporary carbon sequestration may be there by some forest, but the most important role of microorganisms, which are enzymes, to degrade these sequestered carbon. What is sequestered carbon? Cellulose and starch, which is trapped in the form of, what you can say, fruits or plant material itself, all this. So once carbon is active in the atmospheric cycle, it is difficult to lock. Locking up can be done only by growing a plant, or let us say creating some biomass, a biomass like algae. Let us say algae is grown, plants are growing, humans they are developing, population of human is increasing, that traps the carbon. So this is the carbon and the role of starch. So where does starch fit in the carbon cycle? If you see the photosynthesis, from the sun energy photosynthesis, sugars are formed which can be later converted to starch, fats, proteins, etc. by respiration, and then when that animal dies, then suddenly that is what you can say, released back into the atmosphere. So starch is one important carbon source synthesized by plants during the carbon cycle, that is excess. When there is excess of carbon in the sink, it starts to store it as reserve food material. Now there is enzymatic hydrolysis of starch. Where can you find it? You can find it in our body. In the mouth we have saliva, in the intestine we have enzymes. Many microorganisms like bacteria and fungi, they have enzymes specially to degrade the starch. Now, so now we will be seeing the degradation of starch and how that plays a very significant role in carbon cycle. That is what I said. So abundantly it is present in soil, and that is added in soil, present in soil, grown in soil. So soil bacteria, they are very efficient in degrading the starch. There are at least 3 different enzymes and 3 different mechanisms used by the microorganisms to degrade starch. It is not that always when a starch will be acted upon by enzymes it will form glucose, but intermediate compounds will also be formed. These organisms which have the capacity to degrade the starch are called as saccharolytic organisms or amylase producing organism, because amylase is the enzyme which is responsible for degrading the starch. These amylase producing organisms are very common in soil because their natural habitat is soil, and plants natural habitat is soil, and the plant reserve food material is distributed throughout the plant kingdom. So starch degrading microorganisms as compared to cellulose, they are very easy to cultivate. Cellulose degrading bacteria, they are, or the fungi, they are very hard to cultivate, unlike these are very easy. So starch, as I said, it acts as a sink for the carbon during the day and source of carbon during the night. Okay, it is a sink, that is a reservoir during day, and during the night period that reservoir gets utilized, means during the day earning savings, and at the night spending. So night spending of carbon takes place. In the daytime, the carbon dioxide which is fixed, it forms sugars, it is exported to all the tissues. All these tissues, they are heterotrophs. Only the leaf, it is autotroph, let us say. Roots, then shoots, all these heterotrophic tissues, or immature leaves which are pale green in color, they also need some energy. So the matured leaves which are synthesizing sugars, they will send to all parts of the body for storage, for assimilation and growth. At night period, this starch is converted and consumed to provide carbon source for the growth of the plant. This utilizes the Calvin cycle. We have all seen that in our metabolism studies. Okay, so this is how the carbon sink it functions. So the point of discussion here is the polysaccharide starch. As you can see here in the picture, the tuber, potatoes, radish, then even various types of, all types of tuber, beetroot, they accumulate the carbonaceous compound in the form of starch. We will call it as polysaccharides because it is a multi chain of glucose. So polysaccharides, they are large polymers, as we saw in the last lecture, composed of tens to thousands of monosaccharides joined together by glycosidic linkages. The three most abundant polysaccharides are starch, glycogen and cellulose. Cellulose and starch, these are the two important polysaccharides which are most abundant in nature, and they serve a variety of functions. The main function of starch is storage material, reserve food material. Soil is a natural depository for starch and cellulose, and therefore starch, it forms a key component in the carbon cycle. What is starch? If you see starch, unlike the cellulose carbon, which is mostly linear in nature, starch is a branched polysaccharide. It has alpha 1,4 linkages in the linear chain and alpha 1,6 alternate linkages in the branching chain. So this branching straight chain, it is called as amylose, and the branched molecule, it is called as the amylopectin. So linear molecule of glucose is called as amylose. There we called it as cellobiose. Here we are calling it as amylose and amylopectin. So starch, it is a mixture of two polymers, amylose and amylopectin. Natural starches, they consist of 10 to 30% of amylose and 70 to 90% of amylopectin. Amylose is a linear polysaccharide. It is composed entirely of D-glucose units joined together by alpha 1,4 straight linkages. This is the alpha 1,4. This is the first carbon, second, third and fourth carbon. First carbon and fourth carbon of adjacent sugar molecules, they form a bond and alpha 1,4 linkage. So there are enzymes specific for these linkages. Okay, and when they are broken, it forms dextrins. So dextrins, they are glucose polysaccharides of intermediate size, 2, 3, 4, 5, whatever it may be. So the complete hydrolysis of starch, the branching is due to alpha 1,6, means two branches, cross linking is there with alpha 1,6, means the first carbon of the top chain and the sixth carbon of the lower chain, they have a linkage, cross linking. The complete hydrolysis of starch, it yields in succession glucose. The successive stages are starch, it forms dextrins, dextrins to maltose. Dextrins, these are 2, 3, 4, 5, 6 units of glucose joined together with linkages. You don't have linear only, means there can be two molecules with alpha 1,6, there can be 3 and 2 with alpha 1,6 and 1,4 both. So that is dextrins. When it is much more, even brought down to 2s, it is called as maltose. Then there is a third enzyme which will convert maltose to glucose. So this is a multi-step reaction. 2 to 3 steps of reaction are needed for conversion of starch into glucose. In our body and within several microorganisms, bacteria and fungi both, they contain a gene which is responsible for synthesis of an enzyme. All genes are known to produce enzymes. Enzyme is a product of gene synthesis. So there is a gene for synthesis of amylase which degrades starch sequentially into usable glucose molecules. So the hydrolysis of starch into simpler carbohydrates by acids takes place through various enzymes or a combination of the two. The resulting fragments, they are known as dextrins. The extent of conversion of these dextrins is known as dextrose equivalent or DE in biochemical terms. All the scientists who are working with saccharification or hydrolysis of starch, they use this terminology dextrose equivalent, which is roughly a fraction of glycosidic bond or in the starch which has been broken. As I said, in humans only saliva and the pancreas they secrete the enzyme amylase. We have in our system saliva in the mouth and pancreas, they are secreting amylase which can degrade the starch. They are called as alpha amylases and probably the most widely known amylases. They are also synthesized by different types of bacteria, fungi, animal and some plants. The enzymes which break down or hydrolyze starch into sugars, they are known as amylases. They are a group of enzymes and they are present most importantly in the plant microorganisms. Amylases are present in plants also. They are present in microorganisms, fungi, bacteria, etc. So, how does the enzymatic hydrolysis of starch take place? Enzymatic hydrolysis of starch, it takes place with respect to enzymes. As I said, if you see, this is a alpha amylase enzyme which has the capacity to convert starch to maltose. This is dual units of glucose bound together. This maltose will be converted into glucose by gamma amylase or amyloglucosidase enzyme. I will classify the enzymes in the next slide. Here, how the enzymes are key in starch hydrolysis. Starch can be degraded or hydrolyzed by acids also. Acid hydrolysis, but in nature or in the natural systems, hydrolysis of starch takes place. Means we are talking about release of the starch in the form of CO2 back to the environment. That is mostly happening by enzymatic hydrolysis. Means starch is not directly converted into CO2 by microorganisms. They convert it into glucose, and once glucose is formed, you see, glucose is the key molecule in the nature. In metabolism, glucose, everything starts with glucose. It has a EMP pathway, it has to start with glucose. Pyruvate, it cannot be completed without glucose. Okay, TCA cycle. So the key molecule is glucose, which will then be oxidized and reduced to simpler compounds in the end, giving back to the nature. So this is starchy material with alpha amylase and amyloglucosidase or gamma amylase. It converts into two-step reaction into glucose, which in humans it can be utilized in the blood, whereas in microorganisms it will be utilized, or plants it will be utilized for the cellular metabolism. In plants they also have, apart from starch, they can produce sucrose also in some fruits, and this sucrose is also directly acted by alpha glucosidase enzyme to give rise to glucose and fructose. Fructose can be acted upon by another enzyme to give rise to glucose. So ultimately enzymatic hydrolysis of starch yields glucose. The microbial degradation of starch, if you see, or to utilize the starch, microorganisms, they must have enzymes which catalyze the hydrolysis of alpha 1,4 glycosidic bonds which are formed between the D-glucopyranose residues. Now enzymes which are capable of catalyzing the hydrolysis of alpha D14 linkage are called as amylases. As I have been saying, they are saying that they are widely distributed throughout the nature. Okay, so the classes of enzymes, if you see the classes of enzymes, there are three classes of enzymes: alpha amylase, beta amylase and gamma amylase. These amylases, these are responsible for degradation of starch. Collectively in a consortium they do that. Alpha amylases, they are mainly found in animal, plants and microbes. Beta amylases, they are found in plants and microbes but not in humans or animals, whereas gamma amylase is found in animals and microbes but not plants. Okay, so you can see the various optimum conditions needed for their action, but let us see one by one what is alpha amylase. Alpha amylase is a type of amylase which breaks down long chain saccharides, and it acts on random locations on the chain. Means it can act on 1,4, it can act on 1,6 at not regular intervals. Means if there is a long chain at the first, 4th, 6th, 8th, anywhere it can act, yielding different compounds. Depending upon how many glucose units are there, it will be called as maltotriose, maltose, amylose, maltose, glucose, maltodextrins, limit dextrins, etc. So these are how many glucose molecule in what fashion is there. There is 1,4 and 1,6. So you just take a picture and randomly cut it. Whatever you get in your hands, that will be called as these sugar molecules. So as it can act anywhere on the substrate, the action of alpha amylase is faster than the remaining two enzymes. Okay, so Beta amylase is the second class of enzyme present in fungi, bacteria, as we have seen, but it is not present in humans. It catalyzes the hydrolysis of second alpha 1,4 glycosidic bond, working from the non-reducing end, cleaning up every time 2 glucose units. So whenever there is beta amylase acting on starch, it will give rise to maltose, because as they belong to glycoside hydrolase family, they are well known hydrolases. The best example of this is ripening of a fruit. When a fruit ripens, beta amylase in the fruit, it breaks the starch into maltose, and it results into the sweet or the ripe flavor of the fruit. There are many microorganisms which produce this beta amylase to degrade the extracellular starches when they encounter it in the environment as a source of carbon or in the growth media. Animal tissues do not contain beta amylase. The last one is amyloglucosidase. So the conversion of starch to glucose when it comes to laboratory or purely biochemical method can be done in 2 steps. One is add alpha amylases, then add gamma amylases. Gamma amylases are also called as glucosidases, alpha 1,4 glucosidases or amyloglucosidases or glucoamylases. They have many names depending upon the action. All actions, they specify only one thing, that gamma amylase results into formation of glucose. It acts on alpha 1,6 as well as 1,4 both. So when there is a action on the both bonds, what it will result into single units of this polymer or polysaccharide, and that is called as glucose. So gamma amylase is also a significant enzyme in degradation of the starch. So microorganisms which degrade starch, they are abundant in nature. They include both fungi and bacteria, many of which grow under extreme pH. Means we have thermophilic amylases, we have alkalophilic amylases, and there is not a single habitat which may not contain the amylase containing microorganism, as there is not a single habitat which may not inhibit cellulase for containing fungi. Just like that, fungi also, they have the potential to produce industrially important amylases, and for degradation of the starch to glucose, just like cellulose to glucose, starch to glucose, carbohydrates to polysaccharides to carbohydrates under high temperature, high pressure and varied pH. This process is exploited in the industries. So industrially significant amylase producing fungi and bacteria, they are being used, products are being formed. So we consume those products, we defecate and we urinate, and all this is released back into the environment. Or maybe some those products, they are just degrading, when the expiry date is over, they dump it on the soil, and that also gets released in the form of CO2. So microorganisms which are there involved, they are of wide range. Many microorganisms, they produce this enzyme. Most commonly which are found in the soil or used for industrial production, they are Bacillus subtilis, Bacillus licheniformis, Bacillus amyloliquefaciens, Aspergillus niger, Aspergillus oryzae, Penicillium species. All these microorganisms, they break down the starch with the help of a gene which can produce amylase, and they can secrete this enzyme extracellularly and convert it into glucose and maltose and use them for their own cells metabolism. These are the microorganisms which are involved. So Bacillus amyloliquefaciens, Bacillus coagulans, Aspergillus, Penicillium, Mucor, Neurospora, etc. And this is a list of fungi and bacteria. As I said, you can just pause this slide here and go through the list. Just pass it through your brain and you will find that Bacillus species amongst bacteria dominates the enzyme production amylase. So finally, if you just see the summary of starch and its role in carbon cycle, as I have told you, it is synthesized by plants during carbon excess. It is stored as a reserve food material in the plants. So you form fruits, vegetables, tubers, in the roots, leaves, it is stored. There is, what is the fate of this stored carbon? One day or the other, you eat it, or the plant is dead and is added into the soil. The fate, one day, the fate of this reserve sink of carbon stored in the form of cellulose or starch is hydrolysis, degradation. How does that how does that degradation takes place? By enzymes. Enzymatic hydrolysis of starch by microorganism is the key step in the global carbon cycle. It is abundant, that is the quantity of microorganisms capable of synthesizing this enzyme amylase is abundant in nature, and they can degrade starch by three distinct mechanisms: by alpha amylase, beta amylase and gamma amylase. They use all these three mechanisms, that is the humans, animals, plants, microbes. All these four living systems, I can say all the Monera's five kingdom concept, all these, they degrade this starch and either they store it, form a carbon sink, or release it, that is they sequester carbon or release carbon to back into the environment. Okay, starch acts as a sink for the carbon during the day and as a source of carbon during the night period. So day and night, these plants, they are sequestering and releasing the carbon as shown in this figure. So this is all about the carbon cycle with respect to the starch. Starch, as you can know or understand, is a very important source of energy for humans. Just like as in plants cellulose is stored, we store, see, just think about a different type of sink. You have huge godowns of wheat, huge godowns of rice, you have huge godowns of fruits and vegetables which are being harvested daily. You consume them, that becomes a part of your body. Population is increasing. All this is acting as a carbon sink. Now what we are doing, we are using biofuels. Nowadays if you see Brazil, countries like Brazil and up to a great extent even in India, we have diesels mixed with ethanol. That ethanol is being synthesized from agricultural products, starchy materials. Cellulose is being exploited as an important constituent for biofuel production. So what happens if I am using cellulose or starch for biofuel production? I am putting it in my vehicles and driving my vehicle, I burn the ethanol. So I am burning starch, I am burning carbon. So when this thing happens, the carbon dioxide is released back into the environment. But with human activity, what I just said, you think about the sink, the storage is becoming more and more or more less, and the release is becoming greater. So we are artificially carrying out agriculture, but it is not forestation. It cannot be called as forestation because the life of the plants in the agriculture is very short. As you compare to a natural forest growing, it takes years and years and it sustains a huge ecosystem. Animals grow there. There are wild animals, birds, there are inside the soil, there are insects, flora and fauna, they grow, they sequester the carbon. Agriculture doesn't do that. On the contrary, we are imbalancing this carbon cycle to a great extent. All the countries of the world, the superpower countries of the world, now have shifted their focus from nuclear power to carbon balance. Think about it, it's a great initiative. Means each country has a responsibility to maintain the carbon balance. Dear students, let me tell you, in your college, the NAC committee which had made a survey, they had asked what is the contribution of your college in carbon balancing. Believe me when I said, for first time some 10 years back when they asked this question, we were surprised. We didn't know what to say. But the question was directly related with two things. How much plants do you maintain in your campus? Is your campus green? And how many plants are there which have the capacity to fix carbon? And secondly, how many vehicles are there? If you are bringing the vehicles daily, they had recommended you should at least have one day as a no vehicle day. Ride cycles. Okay, riding a cycle will also release CO2, breathing, but that is totally different. Its rate is slow. Whereas driving a vehicle will also release carbon monoxide or methane or different gases, carbonaceous gases. But those gases are not good for the plants nor for the humans. But let us consider this as carbon dioxide. We will call this generally carbon dioxide. Even if you consider carbon dioxide as being released, the amount of release is exceeding the amount of storage, the sink which I mean to say. And this has resulted into climate change. One of the main aim of studying this paper is to understand climate change, global warming. Global warming and climate change, they are a result of these elemental cycle disruptions. Nitrogen cycle is the next in the line of study. Carbon cycle is primary. Nitrogen cycle is the second important one. Carbon is to nitrogen ratio in the environment plays a key, what you can say, statistics in maintaining a good atmosphere for longer durations. So as students, you have a great responsibility on your shoulders to think over these aspects, to become better citizens, and to contribute to the nature as good as you can. So I hope you understand the depth of this subject. Study it once again. Try to understand the fine points of this edification, this syllabus which has been designed for you, so as you can understand it for an applied fashion. You have to apply it in your life. Having said that, we will be venturing into nitrogen cycle in the same line with the same aim and the same motive. So till then, study hard, try to understand, be more knowledgeable, be wise, be intelligent. Goodbye for now.